Right. Hi there. Welcome to today's webinar. Today we're talking about plant metabolism, and this is a special focus collection for plant physiology. And today we have three speakers who have papers submitted and published in this focus collection. Pengda Ma, Tony Grube Anderson, and Hiroshi Maeda. We also have one of the editors of the Focus collection who's going to be giving us a little overview of the topic. That's Spongile Mafu. And we have a moderator who is an assistant features editor for plant physiology, Yushen Fan, who will be introducing the speakers and handling the questions at the end of each of the talks. So I just want to mention that Yushen is a postdoc, and she did her work mostly in Australia at the Australian National University. She's done her fair share of lab hopping, but one thing has not budged: her unwavering passion for C4 biology. She also has a first author paper in this collection on deal-driven variation of leaf dark respiration and metabolite levels in C3 and C4 grasses. So I will be adding links to the chat as we go along so you can find the focus collection and some of the articles in it. What I will do is I will quickly point out that this celebration of plant physiology is online. You can find the focus collection. It was edited by Alain Goussin, Li, Spongile Mafu, and Yang Zhang. And thank you very much for the work that went into this. We will be posting this recording to YouTube, and we have a whole bunch of, where'd it go? Previous webinars on the YouTube channel. I'd like to thank ASPB members for supporting this project. And if you're not an ASPB member, please consider joining. I know it's a bit late because we start in slightly less than 2 months, but the plant biology meeting is held this year in Canada in conjunction with the Canadian Society of Plant Biologists. If you can't make it there in person, we also have a virtual option where you can see most of the talks online. Please put questions for the speakers into the questions box. And again, here is our program today, and I'm going to go ahead and turn this over to our moderator, Yushen.
Thanks, Mary. Good morning, afternoon, and evening, folks. My name is Yushen Fan, as introduced. It is my honor to moderate this webinar, and it's also my honor to introduce you our host today, Spongile Mafu, who's a reviewing editor at Plant Physiology. She's also an assistant professor of biochemistry and molecular biology at University of Massachusetts Amherst. Her research centers on terpenoid metabolism with a focus on biochemical and genetic mechanisms that give rise to chemical diversity in plants. Her work offers insight into how specialized metabolites shape plant defense, environmental adaptation, and ecological interactions, with relevant to sustainable agriculture and biotechnology. So now I'm going to hand over to Spongile for some introductions of this webinar. Thank you.
Okay, hello everyone. Sorry about that. So thank you for the introduction. And I just want to start off with acknowledgements to everyone. So thank you, Eugene, for the introduction, but also thank you for moderating the session. And thanks for Mary for organizing the webinar. And our image was generated by Dr. Sun from Tennessee State. And to all the speakers, we are looking forward to hearing about your work. And all the researchers that made the focus issue possible and a success, and to all of you for joining us today. Okay, so the focus issue was initiated at the end of 2024. And as mentioned, there were the 4 editors, Alan, Lee, myself, and Yang. And it is culminated with groundbreaking articles covering reviews and updates, exploring the multifaceted roles of plant metabolism. And the collection shed lights on the biosynthesis, regulation, and the diverse functions and engineering of plant metabolites. And I will give a highlight and summary of the subtopics covered in the issue, or how the submitted articles have turned out. And I'm acknowledging that all the images displayed in the coming slides are excerpts from the updates and reviews and the research articles, and I'm unable to include everything that was submitted to this particular focus review. Okay, so we started off a central theme of metabolism is also going to be plant primary metabolism. And that turned out to look at the core mechanisms, the regulatory networks, and also the agricultural implications. And so there's an update in that particular issue that then it does a very good job of highlighting the importance of primary metabolism in the metabolism landscape, and they look at the importance of photorespiration. There are also research articles that then went on to show the applications or the influence of primary metabolism for water nitrogen use efficiency and regulation in maize productivity. And Eugene's paper that also then looked at the diurnal shifts in leaf stalk respiration. And the Maeda group, and I think that's what Hiroshi will be talking about today, is looking at the evolution of the aromatic biosynthesis. So our second core theme that came out from the submitted updates and reviews was then on specialized metabolism. And these research articles then highlighted how integrated networks shape plant development and adaptation. And just a highlight of the papers, one of them was looking at plastid remodeling, looked at Stay Green, and looked at plastid remodeling acting as driver of pigment metabolism. There's a set of papers that then also really looked at fruit ripening for example, in tomatoes shown here, but also in cherries. And what they highlighted was the integration of hormones, so primary metabolism, and its influence on developmental regulation and also fruit metabolite quality. And I think what those papers also highlight is the translational effect of understanding plant metabolism in defining or really being able to enhance fruit quality. There's a section of papers also in this particular session of the specialized metabolites that looked at lignin and phenylpropanoid metabolism, and it looked at it at the interface of development and at the interface of development and stress biology. And so that then summarized the specialized metabolism section. Peng Damar then today will highlight specialized metabolism in response to abiotic stress and hormones. And so we'll be looking forward to hearing about that. So we had several updates that were submitted, as well as the reviews and a few papers. I think what they all do a good and awesome job is summarizing the updates of the field, the diverse all the diverse metabolite classes that we have, and then also that the contributions have this unifying theme of plant metabolism as an integrator of function, of being able to dynamically coordinate development, defense, and environmental interactions. And then the emerging challenge is just not cataloging our metabolites, but to define their roles in controlling specific biological and processes and physiology. And so the updates really do a good job of showing what the metabolites do and how that is translated into their biological functions and their importance in plants. And today we will be hearing from Tony, whose topical review synthesizes decades' work of Arabidopsis thaliana and illustrating how the plant metabolites are not really just static products, but are dynamic and spatially organized and environmentally responsive chemical defense metabolites. And then lastly, as we all know, one aspect that has pushed the field further is really all the technologies. And so there are a couple of updates that then emphasize the importance of plant metabolomics from Amit Rai, who then showed the importance of metabolomics in studying plant metabolism. And then also coming from another review highlighted the single cell RNA-seq and metabolomics in the importance of studying the metabolism. And then there's also a nice paper that's in there that really looks at how they used omics to make to compile a library of non-coding RNAs, and it really just emphasizes the tools that then enable advances in research as well as a resource to the community. Okay, so collectively, the updates, the reviews, and the research articles reinforce metabolism as an integrated and dynamically regulated component of plant biology, and they are governed by interconnected transcriptional, hormonal, redox, and developmental processes. And then they also identify shared regulatory nodes across pathways and contexts, not only as advances in conceptual understanding, but are also essential for harnessing metabolic diversity to improve crop resilience, productivity, and quality. And so we look forward to hearing from all the speakers, and thank you, and I hand it back to Yushin.
Thank you very much. It's funny, right? Right. So let's get it with today's courses. The first speaker of today, Honda Ma, who is a professor and a PhD supervisor at College of Life Science, Northwest Agriculture and Forestry University. He was a visiting scholar in UK, two organizations, joint centers, and also University of Glasgow. His research is dedicated to the regulation of secondary metabolism in Salvia malteolorhiza, which I believe is a very popular medical plant called red sage. And that's where he established comprehensive regulatory networks for jasmonic acids and salicylic acid. I still can't pronounce all the metabolites well. In relation to active compounds, Dr. Ma has published a large number of papers in prestigious journals, and today he will talk to us about his latest research article in this special issue about DOF32 coordinates salvianolic acid biosynthesis and drought tolerance in red sage. I'll pass it to you, Honda, if you want to share your screen.
Okay.
Excellent.
Can you hear me?
Yes,
Okay.
we can hear you. Excellent.
Okay.
Thank you. We can see your screen too.
Okay. Hello, everyone. Thank you for being here. My name is Peng Ma, and I'm from the Northwest Agriculture and Forest University. I want to talk about how a plant called Salvia multiorchivum controls its medicinal compounds, especially through a key gene we find called DOF32. To begin with, what exactly is Salvia multiorchivum? Known globally as a red sage or danshen in China, it is a plant with a remarkable story. The name itself reveals its secret. Dan means red, pointing to its vivid red root. The shen means available medicinal root, much like ginseng. But technically, it is a perennial herb belonging to Lamina family. historically as recorded in Compendium of Materia Medica in 1596. These red roots have been used for centuries to invigorate the blood. Today, modern science reveals this power comes from the specific medicinal compounds, tensional and sulfonolic acids. Because these compounds, salvia multi-alcohol is now widely used globally to test cardiovascular diseases. Our research focuses on unlocking how plant synthesizes these crucial metabolites. How does plant make these? They come from the different pathway inside cell. Tensional come from the terpenoid pathway, and the sulfonolic acids come from the phenylprotopy pathway. It looks complex, but plants know exactly how to balance them. Over the past few years, our lab has systematically decoded this jasmonic acid-mediated regulatory network. It starts at the top with the GAZ repressor, which we found exhibit complex functional diversity in regulating metabolism. When jasmonic acid triggers their degradation, it releases the cofactor MYC2. MYC2 then physically interacts with MYB36. This is MYB36 acts as a crucial metabolic switch. It directly regulates structure gene and indirectly moderates factor like ERF6 and ERF11 strongly promote tensional while suppressing the sulfonolic acids. Beyond transcription, we uncovered a post-translational layer, CSN physically bind to the MYB36, preventing its degradation and stabilizing this metabolic shift. Operating in parallel, the SAP4 module, activity by upstream factor, also drive this dual metabolic regulation. In short, jasmonic acid signal orchestrate a hair coordinate multi-layered network to precisely balance the biosynthesis of these specific medicinal compounds. Now let's look at a very interesting problem we call stress metabolism dilemma. In nature, if the drought is severe, it damages the plant and threatens its survival. However, if the drought is moderate, it actually acts as a positive signal. It pushes the plant to make much more medicine, like tensional and sulfonolic acids. So the big question is, how does plant find the perfect balance? How does it survive the stress while spending energy to make these active compounds? This balance is the black box we want to open. To find the answer, we discovered a responsive gene called DOF32, as you can see from our expression data here. DOF32 is highly sensitive. It reacts rapidly to both jasmonic acid and drought signals. To test its function, we overexpressed DOF32 in salvia. The transgenic plant show a remarkable robust, drought-tolerant phenotype with enhanced ROS scavenging capabilities. At the same time, these plants accumulated significantly higher level of sulfonolic acid. This clearly indicates that DOF32 successfully bridges stress adaptation and metabolic biosynthesis. To determine exactly how DOF32 achieved this, we first needed to find its direct targets. By combining the DAP-seq, RA-seq, we successfully located onto two structural genes, LOX8 and RS1. We then validated these multi-omics finding biochemical assays, including EMSA and DURLAC. The result confirmed that DOF32 directly bind to the promoters of these two genes. By activating them, the DOF32 significantly pushes the biosynthesis and accumulation of sulfonolic acids. Because LOX8 is a direct target of DOF32 and a key enzyme in the jasmonic acid biosynthesis, we independently checked its function. Overexpression LOX8 significantly increased endogenous jasmonic acid levels, accumulated more sulfonolic acids, and improved drought tolerance. This reveals that DOF32 forms a positive feedback loop by upregulating the LOX8 to amplify the jasmonic acid cascade during drought. Finally, to complete this signaling cascade, we trace the upstream mechanism. We identify the transcription factor MYB97 directly activates DOF32 expression. Crucially, the core jasmonic acid repressor, the JZ8, interacts with MYB97 and represses its activity. This brings us to our final model. When drought hits, the jasmonic acid signal releases MYB97, which turns on our manager, DOF32. Then DOF32 does two things. It pushes the plant to make much more sulfonolic acid and for protection, and it creates a loop to make more jasmonic acid. This loop makes the whole defense system even stronger. It's a perfect way for the plant to balance survival and medicine production. Now, let's look at the future. Usually people think plant is stressed, so it makes medicine. It survives, but we're asking a new question. Is the medicine itself the reason that the plant survives? For example, do these chemicals can act as bodyguards that clean up the damages for drought? We want to find out if the medicine is the cause of the resistance, not just a side effect. Our next step is build a full system. We want to see how upstream genes control the metabolites and how those metabolites change the physiology of the plant, like fixing the cell wall or cleaning the oxygen. This is not just for the science, it helps us to grow better. Stronger medicine in the field by using the plant's own natural logic. Finally, I want to thank my team at Northwest Agriculture and Forest University. I want to thank my professor and all the students who worked hard on these projects. I also thank the NSFC and other foundations for their support. Thank you very much for your time. I'm very happy to answer any question you may have.
Excellent. Thank you very much, Pengda. So we will have we'll have time for one or two questions. It may take some time for the audience to type in, so I will start with my very first burning question for Pengda. So your data suggested SM-DOF32 directly regulates both SM-RAS1 and SM-LOX8, if I understand correctly. Do you think the DOF32 functions mainly as a metabolic coordinator, or it is part of a broader stress integration network? So does it start, or it is just one of the bigger regulation network that happens to show up?
Sorry, my English not well, so I'm not very clear your question. Can you
Right.
pardon?
Sure. So what I'm thinking is, do you think DOF32 affect RAS1 and LOX1 directly, right? Do you think it's because it is a metabolic coordinator? So DOF32 somehow activated and start telling RAS1 and LOX8 what you need to do under drought stress? Or do you think DOF32 is part of a very big, broader stress integration network? So it does not act, how do I say, individually or independently, but as part of the bigger network. So,
that's a good question, because in this work we only find that the DOF32 is a master factor to affect the drought tolerance and affect the metabolism. That's all. But we don't know the reason medicine maybe affect the drought stress. We don't know. So next step, we will find the reason or not.
Okay.
Yeah.
Okay.
Yeah.
Excellent. Sorry, I'm really terrible at explaining things. We do have question coming in. So a question from Rob. How did you identify SM-DOF32?
Where?
I read it out for you. How did you identify SM-DOF32?
How you mean the how identify?
Yes. How did you identify?
We screening the transcriptome, and we find the DOF32 and react both the jasmonic acid and the drought stress is very highly. So we think maybe the DOF32 is a master transcript factor, maybe affect the solvinolic tensional or the drought tolerance.
Okay. Okay.
Yeah.
And last quick question. How was the drought induced? What was the condition?
You mean the condition?
Yes, the drought condition. How did you control the drought?
We use the one is the hairy root. Another is a young shooting, and put some the PEG or something else. Yeah.
Okay. Okay. Great. Folks, I can't see all the attendees, but please join me to thank Pomda
Yeah.
for his talk. Thank you very much. So we're gonna move on to our next speaker. We have Professor Tony Goob Anderson, who is a group leader and professor at Department of Plants and Microbiology at the University of Zurich. He was a Marie Curie Fellow. He was also a Humboldt Fellow, almost a role model for me than my future past. Now he's an independent group leader in Zurich. He works on a lot of things. So if I have to summarize, his research is specialized in figuring out plant defenses, I believe. How do plants know what to take up, and how do they know what toxic or bad things to keep out? We'll have Tony today to give us a topical overview of Arabidopsis chemical metabolism and defense research. Thank you, Tony.
Thank you very much, Yushen. I think I'm sharing my screen now. No, yeah.
Yes,
yeah, thank you very much
for
this very nice introduction. I'm really happy to be here, and also thanks to Mary for organizing all this. It's really a privilege to be part of this series, and it has also been very exciting to be part of the thematical collection. So yeah, I will tell you a little bit about these chemicals and about also some other things in plants that might be interesting. So I titled my talk, "We Will Block You, the Interplay Between Physical and Chemical Root Barriers." So you might sense a little bit of a musical theme in my titles. That's something that's a little bit reoccurring. Let me see. So yeah, so as you said, no, my lab is very interested in a number of different things, and I think you summarized it really well, but we're also interested in, development in a way where things like, for example, this root is really fascinating to us. Plants are really exciting in many ways, but specifically because they really don't have any particular shape. They adapt to their environment, and that's one of the things we are really curious to find out. We have 3 pillars, so to say, in my lab. We are very interested in root development, and particularly in natural conditions. So how do roots actually behave in the soil? And of course, also the physiology that's associated, and specifically the defense chemistry, which we'll get a little bit closer into today. And you can barely say that without saying root and microbe interactions. So we are very interested in these different things. And we have projects in each of these pillars. But overall, if we are to summarize our interest, it is into how roots integrate environmental signals perceived by the individual cells into their growth and function. So we have a number of different things that we study in these aspects, and we have in the lab 3 supermodels we use. We use in particular Arabidopsis thaliana. It's rather useless, but when it comes to molecular mechanistic insights, it's just simply unbeatable. I'm sure I don't have to tell anybody that many of the things, almost most of the fundamental molecular insights we gain come from this plant. We're also starting to translate these things into agriculture, and we're using a crop, in fact, which is called Camelina sativa. It's a very nice crop. You can actually transform it by dipping. So we started calling it Arabidopsis 2.0 because it has many of the same advantages that you have in Arabidopsis. My lab is also interested in spatially constrained plant microbe interactions inside nodules. So we have another plant species that can form these very interesting structures called nodules, and this is Lotus japonicus. And this is very exciting. I don't have time to go into that, but today we focus on Arabidopsis. So I'm going to tell you a little bit about this. And then the reason I'm here is because we wrote this review together with Dan Kliebenstein and Mark Somsic. This was really a very interesting experience and very cool. No, we wrote this because we decided, Mark looked into what was known about the defenses within Arabidopsis and their actual function. So we decided to write a little bit more, what do the chemicals actually do in Arabidopsis? So if you read this, you'll see that this is more focused on the actual output of immunity and how defenses actually work against different microbes. So we summarize this with this figure, which also thank you for including in the introduction, where we have a little bit of an overview about the major different classes that occur of defense chemicals that occur in Arabidopsis. And today we're going to focus on a particular branch of that with an example of another project that we work on, which is focused on these particular compounds, which we try to call tryptonides. So these are all tryptophan-derived metabolites from Arabidopsis. And tryptonides are quite interesting because these are these are really the weakness of fungi, so to say. These are really known as fungicides, so they're really efficient in fighting fungi. So the plant is using these as really a defense chemical against fungi. There's a number of different ones of these. So most famous are the indolic glucosinolates and camelexin, which works as inducible defenses in Arabidopsis. So once you have PTI or ETI activated, you also have downstream production of some of these components. There's also a number of other unknown tryptonides that are happening in Arabidopsis. There's a lot of, particularly CYP71 class of P450s that are doing unknown things. So there's a huge potential for discovery of new compounds even in Arabidopsis. But what we were particularly interested in this case was how do these tryptonides actually work in the roots? So my lab is very underground focused. We really like to focus on the roots. And here in this project, we were interested in a tissue known as the periderm. So the periderm is a very interesting tissue that occurs in the older parts of the roots, and it's a protective tissue. So it's really serving as, you could say, the armor of the plant. It's really ensuring that the root is very covered and really secure. So in order to study this, of course, we want to get rid of it and see how the plants respond. And a couple of years ago, we published a mutant where, for example, you can see here on the left side is a wild type that's stained for suberin, which is part of the barrier, the physical barrier in the periderm. And then you can see here a mutant where we were able to completely get rid of that. So we could essentially make a plant that has lost this protective ability in the old parts of the root. And this is where at that point in time, PhD student Pascal joined. He now graduated, so now he's a postdoc, and he was interested in figuring out what comes out of when the periderm is disturbed. What happens in this plant, and is it capable of surviving? So what Pascal developed together with our collaborator Sandra Jemgar from UMO was a system where he was able to actually detect exudation across different zones of the root. So in the end, what they did was they 3D printed a square plate with small different 1 by 1 centimeter sections they could fill up with agar, and then they could position plants onto this. and thereby allow the root to exudate into the different zones and then collect this from the different environments and then analyze what comes out in different parts of the root and correlate that back to the different root parts. So here it's just an illustration of an untargeted analysis where we see different features that pop up in different parts of the root. So you can nicely see that there is a change of the identity of the features from the top of the root, so where the periderm is, down to the tip parts of the root. And this is very exciting. So there's a lot of things to show here, but just very quickly, what we could actually find out is that in a wild type, these tryptonides are produced just below the actual physical barrier that constitutes the periderm. And when we disturb this, when we remove the armor of the plant, the tryptonides take over and compensates. In fact, they're upregulated when the physical barrier is destroyed. So we think there is a compensation effect here where the physical and chemical barriers are interacting. And interestingly, at the same time, another PhD student in a neighboring group was studying a fungi called Plectosporella cucumella. And what she found out is that, when we looked at her infections, we could see that they were really biased towards the periderm part of the root. And here you can see a cross section of a root that has a periderm. You can see the fungi in half, and you can almost feel how the tryptonides are sitting just below here and keeping the plant out from the inner part of the roots here. So of course we wanted to investigate that a little bit deeper. So what we did was we looked at the infection and how the plant survives when they are infected by this fungi. And here you can see the plant that has no periderm, and here we obtained another mutant which cannot produce any tryptonides. You see they're both a little bit smaller than wild type, but when we cross those two, you can see that the plant really suffers, and it really gets invaded by this fungi, and it's really not that happy. We measured how much fungi is actually inside the periderm, and here you can see the relative amount of fungi in wild type versus the two single mutants that have either the chemical or the physical defenses. But once we remove both, we have a lot more fungal DNA inside the root. The same in the shoot. We can also see that it gets over-colonized by the fungi in this case. I should mention that these plants were actually grown so the roots were completely separated from the shoots. So the only way the fungi could enter here was by growing. So to summarize what we have found here, I could talk about this forever, but I have very little time. So what we found was that we have this separation of a physical and chemical barrier in the periderm. When we remove the physical barrier, the chemical barrier compensates. And then when we also remove that, then the tryptophan derived or the tryptonides, they are exuded or they come out of the periderm and fight whatever is present. When we remove that, the plant is really suffering and is really open for invasion of different fungi. We have a lot of data also showing that this is actually relevant when you put the plants in the soil, that they really also die when they don't have these defenses activated. So this is something that potentially is relevant to look into from an agricultural point of view. And to summarize, I would like to thank in particular Mark for writing this fantastic review and all this experience that was connected to this. For this project, this was initiated by Julia Holbein and then taken over by Pascal Brun, who both did an excellent job in making these nice data I showed you. Also to Silvina for helping out with all the chemical analysis that we did, and Laura and David for help also with some of the periderm analysis. And of course, thank you for your attention, and I'm happy to answer any question.
Excellent talk. Thank you, Tony. I'll just pretend, I'm on behalf of all the attendees clapping my terrible hands. Right.
Thank
so
you,
we do have two questions coming. So the first one, what is the differences between tryptonides and auxin/auxin derivatives molecules?
Yeah, that's a very good question, actually. I would say, given our definition, auxin is also a tryptonide, right? So we are thinking whether there might be certain kind of development versus defense trade-offs in which direction these chemicals are funneled into. You can imagine if you use, for example, tryptophan to create auxin, maybe you have less chemicals available, but you can use the auxin to induce cell divisions and make a thicker periderm, for example. Whereas if you funnel more into, let's say, camellexin, then maybe you don't get such a thick protein, but you get more chemicals. So we think there might be certain, kind of a balance in that point. But yeah, in principle, the auxin and the derivatives should be also tryptonides.
Right. So we have a second question from Akita. Do you check what happened in case of symbiotic fungal interaction?
Also a very good question. So Arabidopsis and the Brassicalis are a little bit special in that sense that there are really not that many symbiotic fungi that interact with them. And the question is whether this is correlated or whether this is due to some of those specific chemicals that Arabidopsis and the Brassicalis they can make. So it's a very interesting question, and I think there's a very deep evolutionary answer to that. But I think maybe this is something for a later, deeper discussion.
Right. So we do have a number of questions, so I apologize, I may not pick all of them, but Tony will have the opportunity to type in the answer there. So gonna pick the next question. What about the effect of tryptonides on mycorrhizal fungus? Have tryptonides been studied in trees?
So this is also a good question and related to the previous question that some of the tryptonides are indeed made by many different species, in particular more towards the auxin kind. Of course, auxin is very essential for plants, but some of those camellexin and some of those more specialized compounds are actually specific to Brassicalis. So the influence on mycorrhiza is not really known because the plant that can make mycorrhiza, they don't really make those particular tryptonides.
Right. So next one. What kind of specific stress could induce tryptonides accumulation? Or is it non-specific?
So typically these are induced by damp responses, and they're also jasmonate dependent. So they are really canonical in sense of how inducible or inducible defenses are typically responding. So there's nothing. If you have a jasmonate mutant, for example, AOS mutant, it cannot produce these compounds at all. So this is a typical response.
Right. So the two questions almost the same. They're both interested in whether tryptonides are, first of all, a physical defense layer or a chemical defense layer. Whether it is the last line of defense or it's just a starting point of the defense.
So I think the last layer of defense is typically this hypersensitive response or necrotic response where the plant simply kills the cells, right? That's typically considered a last resort. So in this sense, I would say they represent more like a let's try to fight this response. No, they are not sure whether the actual mechanism for how they kill fungi is really known, but there are some fungi that are more susceptible to this than others. And what we're currently actually looking into is to investigate if fungi that are more resistant to these chemicals thrive better in the rhizosphere or in the microbiome of the soil that surrounds these plants. No, because you could almost expect that you would select then for resistant fungi that surround that area.
Right. So we're gonna move on to the last speakers. I apologize once more that I did not pick all the questions in the chat box. But Tony, can I invite you to have a look at?
Yeah,
have a look at the Q&A. Excellent. Thank you so much. All right.
Thank you.
So our last speaker, thanks. Our last speaker is Hiroshi Maeda. Hiroshi is a professor at University of Wisconsin-Madison. I still struggle to pronounce the state's name of all American states. He works in botany and cellular/molecular biology department. He has a very strong connection between Japan and the States, given that he did his bachelor and master back in Osaka and PhD in Michigan State University. So his lab focused on nitrogen metabolism, nitrogen metabolic network and biosynthesis of aromatic amino acids, which are essential for human nutrients and precursors of numerous plant natural products. So the overarching goal of his lab is to find the basic for synthetic biology or sustainable productions of a number of aromatic chemicals directly from CO2 using sunlight energy in plant roots. So today he's gonna tell us about aromatic amino acids in grass lignin. Up to you, Hiroshi.
Yeah, well, thank you very much for the kind invitation.
Thanks.
And also, I'd like to thank ISBB for this great opportunity to present our recent work published in Plant Physiology. This was done by Jorge Elazaz and Bethany Moore in my lab at Wisconsin-Madison. So I'll be presenting on behalf of them. So today I would like to discuss how the primary metabolism, aromatic amino acid biosynthesis, underwent the evolutionary alterations during the evolution of grasses through the studying a series of grass species, as well as some of the related species, such as Joinvillea and forest, that trace back the evolutionary history of this evolutionary change. So plants can capture CO2 through photosynthesis, and you convert this one carbon metabolites into a number of specialized metabolites that are produced in different plant lineages. Many of these compounds are specifically produced in certain plants, therefore called specialized metabolites. But these molecules are derived from common precursor primary metabolites, such as amino acid, that are synthesized through typically highly conserved primary metabolic network. But my labs has been interested in understanding or asking the questions, are there any differences in this primary metabolism? Are there any diversity exists in primary metabolism, especially at the interface between primary and specialized metabolism, so that this difference could be useful tools to engineer plant metabolism in the future. So in today's talk, I would like to focus on this metabolic interface between aromatic amino acid biosynthesis with phenylpropanoid biosynthesis, where the upstream pathway synthesizes these three aromatic amino acids, tryptophan, phenylalanine, tyrosine. And typically, most plants utilize this phenylalanine as a starting precursor molecules to produce diverse phenylpropanoid compounds, including cell wall lignin. And this is mediated by this committed enzyme PAL, phenylalanine ammonia lyase, that direct carbon flow into this diverse network. But grasses previously have been reported to have somewhat unique, different metabolic network in the phenylpropanoid and lignin pathways. But most importantly today, the grasses can utilize both phenylalanine and tyrosine as a precursor molecules to synthesize phenylpropanoids, having this dual entry pathway due to the presence of this enzyme called PTAL, the phenylalanine tyrosine ammonia lyase. And also, in coordination in previous studies, we found that the grasses have somewhat unique isoform of aromatic amino acid biosynthesis enzyme. First, this Ti-A arogenate dehydrogenases that catalyze the last step of tyrosine biosynthesis are typically strongly feedback regulated, but grasses have a specific isoform that are less sensitive to feedback inhibition, therefore can efficiently synthesize tyrosine more efficiently to provide precursor to this PTAL enzyme. Also, the first step of the shikimate pathway, DHB synthase, is also somewhat different. One of the isoform called DHS1B is again escape the feedback regulations. Therefore, grasses, some of the grasses such as Brachypodia can direct more carbon flow into the shikimate and aromatic amino acid biosynthesis. So in this studies, in most recent study in plant physiology, we next asked, so when and how these deregulate DHS and Ti enzyme isoform evolved during the grass evolution. So to answer this or to address this question, we utilized a series of grass species belong to both BOP grasses and PACMAT grasses, two different clade of the grasses together constitute core grasses. But also, we use Phallus latifolius, which is the grass species sister to entire core grasses. And also, the Joinvillea ascendens, shown here, which is non-grass poales or gramineates, that is sister to the entire grasses, which was in collaboration with JGI and Jim Liebensmack and Michael McCain, we obtained the genome sequence of these two species. So we're utilizing a series of grass and related species to study the trace the evolutionary history of this metabolic changes. So basically we ask when and how these unique enzyme isoform evolved during this grass evolutions, particularly in relation to this row whole genome duplication that occurred at the common ancestor of all grasses, and also the evolution of PTAL enzyme that occurred just before the emergence of grasses, which was recently published in the bioRxiv, which is currently under revisions. So first, using these genomes, we built a phylogenetic tree of TIA, and we found that TIA genes duplicated into TIA 1 and 2 isoform in a tandem manners within the core grasses. So somewhat recent duplication occurred. But also, this highly active TIA non-canonical isoforms are present in all monocot species. And when we express all of the non-canonical TIA from different grasses, maize, rice, and brachypodia, as well as Joinvillea, as well as pineapple here, we are able to see that when it's expressed in Nicotiana bentamiana leaves, the level of tyrosine dramatically increased in these infiltrate leaves, which was not the case when we expressed the inhibited TIA enzymes from brachypodia, suggesting that many of these monocot species have active TIA enzyme. Next, to look at the phylogenetic tree of DHS genes, we found that DHS1 isoform duplicated into 1A and 1B isoform, and this likely occur at the common ancestor of all grasses, likely through the row whole genome duplications. And when we then test biochemically, first the brachypodia 1A and 1B enzymes, when expressed again in Nicotiana bentamiana, we found that 1B enzyme expressions led to dramatic increase in phenylalanine and other aromatic amino acid, as well as shikimate, as expected. Joinvillea only had one copies, didn't have duplicated copies, but its expression didn't increase the phenylalanine levels. The Phallus had two copies, 1A and 1B, but even the 1B expression did not dramatically increase phenylalanine or shikimate levels, suggesting that although these some of the grass species have two copies, but not all of the 1B isoforms are active enzyme or highly active enzyme. So to further explore when this highly active DHS1B isoforms evolve, we characterize different DHS isoform from BOP grasses, brachypodia and rice, as well as PACMAT grasses, the sorghum and setaria. And we found that the 1B isoform from BOP grasses, expression of that in Nicotiana again, led to dramatic increase in these aromatic amino acid metabolites and intermediates, suggesting that this 1B isoform became deregulated and became highly active very recently within this specific lineage of grasses. So knowing when these changes occur, now this gave us the opportunity to ask how this happened. Now we compare this structural model as well as the protein sequences of highly active DHS1B isoform from BOP grasses compared to the other PACMAT and monocot 1B isoform, and look for the residues that are alters during this specific evolutionary transition. And we identified 17 different potential candidates. And Jorge has done a number of mutagenesis studies and boiled down to these 6 residues. And when these 6 mutations are introduced into the phallus DHS1B, which itself, wild type, is not highly active, but then this 6 mutation converted this 1B isoform from phallus into a super active enzyme. That expression led to a higher level of phenylalanine accumulation, actually more than the Brachypodia 1B enzymes. Then trace back which residues are further important. He found that the threonine 211 to methionine conversions, it has some effects, but when this mutation is combined with the remaining 3 mutations, now essentially can generate this similar highly active enzyme. And these 4 mutations are located at the putative allosteric binding site of this DHB synthase enzyme, which is far away from active site shown here, suggesting that these mutations alter the sensitivity to the inhibitors, therefore now became less sensitive to feedback inhibition. So putting all this together, in summary, we found that all monocots started with already quite active Thai enzyme that can direct carbon flow into the tyrosine productions. And later on, just before the evolution of grasses, this bifunctional p-tyl enzyme evolved to now direct tyrosine in addition to phenylalanine into phenylpropanoid biosynthesis. And then later on, DHS isoform duplicated, likely at the row whole genome duplications, and followed by Thai 1 and 2 tandem duplications. And then this 1B copies, the genes or the proteins, actually deregulated much more recently within the BOP grasses. Now, these grasses push the carbon flow into the shikimate pathway to efficiently provide both tyrosine and phenylalanine as a precursor molecules to this study. This research highlight that the regulation of aromatic amino acid biosynthesis has been fine-tuned during the different step of the monocots and the grass evolution. So based on these studies and prior studies, we found a number of evolutionary alterations, especially in the regulation of or some of the pathway connections in this primary amino acid pathway, aromatic amino acid biosynthesis. And this is likely due to the coordination with this primary metabolism to downstream specialized metabolites. And utilizing these knowledge and tools, we can now conduct synthetic biology and metabolic engineering to enhance the production of some of the useful compounds. Just 2 days ago, we have our papers, another paper has been accepted in plant physiology, where we utilize this knowledge to produce natural pigment spalins at high levels in soybean. So hopefully you'll be able to look at this in a couple of weeks. So with that, I would like to thank again Jorge Alzaz and Bethany Moore. Jorge did almost all of the work, and Bethany have conducted the phylogenetic and evolution analysis. Jorge is now starting an independent position in Valencia, which hopefully he would continue to do exciting work in his group. And also, I'd like to thank the current and past members of my lab, as well as JGI and Jim Liebensmack and Michael McCain for the genome collaboration in genome sequence analysis, and also the NSF Plant Genome Research Projects for funding this work. And thank you for all of your attentions, and happy to take any questions.
Excellent. Thank you so much, Hiroshi. We do have some questions floating in, which I also have the exact same question. I will read out one of them. So in your PacMed clade, there are 2 species, both of them are C4. In your Bob clade, there are 2 species, both of them are C3. Do C3 and C4 grasses differ in amino acid exudations patterns?
So we have not compared those particular C3, C4. To do that, we have to look for this, characterize the relatively rare minor C3 species in PAC-MUD clade, so that we can see if this is because of the C3, C4 or the PAC-MUD or BOP. So yeah, that's a great question as well. We haven't tested that question yet. Yeah.
Right. So another question. Is there any significant sequence variations in TYRA of C3 and C4 grasses?
Yes, that we don't know yet as well. We have looked at those differences. That is actually interesting that at the biochemical levels, especially this is the biochemical data of how sensitive or insensitive to a feedback inhibition to tyrosine on this TIA enzyme. Maize enzyme is much more resistant to feedback inhibition than the rice. So there may be additional mutations or adjustments on the TIA enzyme that some of the grasses, including C4, again, we haven't carefully compared the C3 versus C4, but there may be additional mutations. But there were no clear single mutations that we could trace. So we were not able to identify exactly in this study. Yeah.
Great. Next question. How did you experimentally validate that the deregulations of DHS1B directly changes metabolic flux through the shikimate pathway rather than simply correlating with the evolutions of aromatic amino acids metabolism in grasses?
Yeah, so one is the in vivo data, implanted data of expressing this enzyme in Nicotiana bentamiana, which we know they do not have deregulated enzyme. But also, we have done the 13CO2 labeling experiments previously, where we measure the rate of the tyrosine and phenylalanine biosynthesis in grasses versus Arabidopsis. And we found that these grasses can synthesize tyrosine more rapidly, meaning 13C labeled carbon get incorporated into tyrosine more quickly. So this provided direct evidence that the presence of this enzyme indeed enhance the carbon flow through the shikimate pathway. Yeah.
Right. Since we talk about this enzyme, do you know about the expression patterns of this enzyme? Are they tissue specific?
Yeah. So interesting that especially this TIA1 enzyme, is highly active in vascular tissues, where p-tau enzymes are also highly active. Therefore, it's likely that these enzymes deregulate or active TIA and p-tau enzymes are particularly important in lignifying cells, where a large quantity of carbon flow is directed into a phenylpropanoid and lignin biosynthesis. So yes, the answer, these specific isoforms are expressed specifically in certain cells. Yeah.
Have you been able to knock these genes regulatory enzymes out or down, regulating them down? Any impact on the plants?
Let me see. TIA1 knockout did not have major effects on the overall phenotype, suggesting that there's still a functional redundancy between them. I have to double check with Jorge, but TIA2 knockout was probably difficult to obtain. That may be a more sort of a baseline TIA enzyme required for protein synthesis, or the tyrosine required for protein synthesis. Yeah.
Right. So a little bit over time, one last question. What is the relationship between amino acids and biofilm formation, especially in grasses?
Biofilm or biofuel?
Well, the word is biofilm. Bio, F-I-L-M.
Okay. I do not know if it's suberin related. Maybe some of the tyrosine may be directed towards. Yeah. Yeah. Sorry, I don't have a direct connections at the moment. I'm happy to discuss over the chat. Yeah.
Okay. So we are 4 minutes over time. Mary, do you have any announcement?
I would only like to say thank you to all of you. There was some fantastic talks. I really enjoyed those. Spongale, I really appreciate your work and the other editors. As you know, being an editor for Plant Physiology Focus Collection is a ton of work, and you've done a fantastic job. And Yushen, really, you've done a wonderful job, especially considering your your wounded hand. Brilliant work. Thank you for keeping us to time. And again, thank you, everybody. Spongale, would you like to say anything?
I'd just like to thank the speakers again for taking the time, and Eugene as well, moderator, and also everybody again who contributed to the focus issue. It was a pleasure editing all of those articles. And thank you again for everyone who's attended.
Thank you. Bye-bye.